Hello everyone. Okay, so I hope you can hear me fine. So I give you 1 minute more so you can go back into your seats. And we're gonna start the second part of the lecture today. Let me share the screen. Here we go. Okay then, so welcome back to the session. And this is the lecture number 6. The previous lecture we have talked about the gross anatomy of the hypothalamus and pituitary gland, and making emphasis also in the portal blood of system, the different nuclei, and the gross anatomy and the development. However, what we have not covered and we will be covering in this lecture is the hormones that are produced. Obviously, as a physiological organ, the final goal of the hypothalamus with the pituitary gland is to actually secrete hormones that would regulate peripheral organs. In turn, these organs have functions such as regulating the metabolism, such as the thyroid, but also reproduction, such as, for example, reproductive organs like the ovaries and the testes, and also, for example, the adrenal glands. Each corresponding axis will be explained in different lectures, but also the disease associated to them. But obviously, to understand the function of this homeostatic control, we need to understand how the hypothalamus, which hormones it secretes, to control the secretion in the pituitary gland. So, as an overview of the lecture number 6, this is what we're going to cover now, right? So, hypothalamic pituitary axis, we're going to cover in this lecture mainly the hormones that are regulated by the hypothalamus, right? So, the hypothalamus is the CNS part, contains the neurons, and it functions as a integrator, right? So, what does it mean to function as an integrator? Just 1 second. So the CNS has a way to get the inputs. These inputs can be either visual or biochemical, or for example, in some animals, smell. For example, some animals, the reproductive cycle is triggered by pheromones. These pheromones travel through the air and smell, they go into the nose, and then it triggers some inputs in the hypothalamic neurons to trigger the pituitary gland to secrete reproductive hormones. Also, other types of inputs can be smells of food, can be visual, and also, for example, the daylight night cycles, for example, that produce melatonin from the pineal gland. It's when the dark light starts in the evenings. So the hypothalamus acts as a CNS center of integrating inputs and will then secrete some neuropeptides that basically will stimulate the production of hormones in the pituitary gland. So this schematic is a must know, right? And it basically shows the importance of the main hormones produced by the hypothalamus. So the draw is very schematic, but what you can see here is The top row is the hypothalamus and the peptides that it secretes, or the hormones neuropeptides, right? We will cover individually one by one. In yellow, what you have is the anterior pituitary. So if we look into the hypothalamus, what we have at the top row is the neuropeptides that the hypothalamus secrete. For example, the first one is GnRH, or gonadotrophin releasing hormone, GHRH, or growth hormone releasing hormone, SS, or somatostatin, TRH, thyroid releasing hormone, DA is dopamine, and CRH is corticotrophin releasing hormone. These peptides or neuropeptides, or as we call it, releasing hormones, have an impact in the pituitary gland and activate certain cell types to secrete certain hormones. In this very brief schematic, what you can see is the action of each neuropeptide or releasing hormone into the cell type that it activates. And it can have two different functions, either activating or either suppressing or inhibitory role. So for example, the gonadotrophin releasing hormone has the function to activate the production of follicle-stimulated hormone and luteinizing hormone from the FSH and LH terminally differentiation, or gonadotropes. The growth hormone releasing hormone has a positive effect into the anterior pituitary to produce growth hormone from the cells which are called somatotropes. Somatostatin has an inhibitory effect, so it suppresses these somatotropes from producing growth hormone. It also has an inhibitory effect on thyroid stimulated hormone, and thyroid releasing hormone has a double effect, positive effect on thyroid stimulated hormone and prolactin secretion. Dopamine has an inhibitory effect on the secretion of prolactin, whereas corticotrophin releasing hormone has a activating effect on the corticotropes to produce adrenocorticotrophin releasing hormone. So this is the hypothalamus and the anterior pituitary. So this is a very depicted summary of the main releasing hormones from the hypothalamus and the anterior pituitary gland. Remember that the hypothalamus is not in contact with the anterior pituitary gland, but these releasing hormones are secreted to the portal vessels, and these are the ones that will contact with the pituitary gland to activate certain hormones and certain cell types in the anterior pituitary. We will cover each one individually, and we will cover the function of each one. So, as an outline, right, of the hormones of the hypothalamus, as I said before, this slide is very basically the summary of all the lecture and is what you guys need to know. But obviously, you need to know also the function of each specific hormone. So if we start, right, gonadotrophin releasing hormone, or GnRH, is a decapeptide. We can see here the structure. You don't need to know this. What is important to know is that this molecule is highly conserved throughout a species and has 60% conserved over the past 600 million years of evolution, and is very similar between human and fish, and human also and sea and queer and siona. So it's highly conserved. So it's a hormone that is being preserved through evolution. Acts the releasing hormone, the gonadotrophin releasing hormone, acts through its receptor, which is GnRH receptor, right? Gonadotrophin releasing hormone receptor. As Peter McCormick taught you in the class yesterday or the one before, these are receptors which are GPCR, and they are found in the pituitary gland and increase calcium, IP3, and PIC kinase. They are key regulators of reproduction. So GnRH will go into the portal system of vessels. It will activate the gonadotroph cells to produce luteinizing hormone and follicle stimulating hormone in the pituitary. And this will go into this main circulation, which in turn will activate the ovaries and testicles for sexual maturation and reproduction. The secretion is pulsatile nature of release, which means that has cycles during the day and is under the control of estradiol and other factors, which we will come into know further on in our module, because there is a specific module on hypothalamic pituitary gonadal axis where we will study all the fertility and menstruation cycle and how LH and FSH specifically regulates reproduction. So again, as some of these hormones have different pulses during the day, and in this case have low frequency pulses, and FSH is released at high frequency pulses and results in also LH release. So where these type of neurons are of high importance, the reason is because they are actually the only type of neurons that are not born in the hypothalamic area, but they are born from the nose, right, during embryogenesis. But more of the nose, the veromesal organ, or what we call as nasal placodes. So what you can see here is an embryonic sagittal section at 11.5, and this is the nasal placode where these neurons are, and migrate into the brain, migrate into the hypothalamus, where then they project axons to the medial eminence and release gonadotrophin releasing hormone that acts in the pituitary. So embryologically, why is this important? Is because the nasal placode that codes for smell also codes for some neurons that will give rise to the reproduction system. So there is a abnormality or disease or congenital abnormality in which lack of smell is associated with infertility, which is called Kalman syndrome. And basically is because the origin of these cells in the nose also are the same origin that produce neurons that regulate reproduction. So in evolution, the neurons that are regulating the smell also regulates reproduction. Okay? And there is a very nice article, if you have time, you can look here in Frontiers of Endocrinology, which summarizes the production of these neurons during development. So gonadotrophin releasing hormone is essential for reproductive development. And using mouse technology, the knockout of GnRH or mutations in the receptor leads to abnormalities of the reproduction tract. So you can see here the testes of a normal mouse and here the uterine with the ovaries, and this is the knockout of gonadotrophin releasing hormone. You can see hypoplasia, severe hypoplasia of the reproductive organs. Here you have another picture. This is the uterine horn, and the ovaries of a normal mouse, and next to it of a mouse mutant for gonadotrophin releasing hormone. In 2009, not that far, right? It was identified that mutations in GnRH1 mutations lead to familial hypogonadotrophic hypogonadism. Hypogonadotrophic hypogonadism is infertility due to abnormal secretion of neurons, of gonadotrophin neurons, into the hypothalamus. So that was the first paper identifying mutations in GnRH and led to a New England Journal of Medicine. From then on, as I will explain at the end of this module, in the reproduction and fertility, there have been many genes associated to gonadotrophins and the GnRH neurons pathway. But we will cover this at the end of the module in May. What is important to know is that the hypothalamus secretes GnRH and that these neurons don't come from the hypothalamus but come from the nasal placode. And they regulate organ development by activating the gonadotrophs in the pituitary gland, which in turn will activate the formation of the gonads. So this is the paper. If you have time and you are interested, you can download it, but it was a milestone in reproductive biology because it's the first identification of GnRH1 mutation in familial, so genetic predisposition to infertility, or also known as hypogonadotrophic hypogonadism. So obviously GnRH and all the releasing factors act through a receptor, and in this case is a GPCR receptor, and it does not have acetyl. We can see here 1, 2, 3, 4, 5, 6, 7 transmembrane domains, and it's found in the somatotroph cells. Now, mutations in this receptor have been also identified and lead to disease such as infertility and hypogonadotrophic hypogonadism. So, what about growth hormone releasing hormone, right? This is another neuropeptide, peptide hormone. This is the sequence, right? You don't need to know about the sequence. This is just to give you an example of how big it is and the composition. It's also quite preserved through evolution, and it's produced by the growth hormone releasing hormone neurons in the arcuate nucleus of the hypothalamus. Remember, the hypothalamus had a specific nucleus. So growth hormone releasing hormone is produced by the ERC arcuate nucleus. The function of the growth hormone releasing hormone is to stimulate the secretion of growth hormone in the anterior pituitary, again by the growth hormone releasing hormone receptor. As Peter McCormick explained you yesterday, these releasing hormones act a lot of times or majoritarily through GPCR. That's why it's very important to know about GCPR receptors. And it leads to activation of adenyl cyclase and cyclic AMP and the transcription of growth hormone. So, in turn, the activation of growth hormone is important, as we will see in further lecture, for linear growth. And what is linear growth? This is us growing in height. So it acts on the bones and the cartilage and the growth cone for us to be growing, but it's also very important in metabolism. Growth hormone releasing hormone mutations were the first identified as an isolated growth hormone deficiency, and it leads to severe dwarfism. And in genetics, a diagnosis is very important. So, what about the growth hormone axis? So, growth hormone is produced by the pituitary gland, but is activated by the growth hormone releasing hormone. The growth hormone then it goes in the circulation and activates IGF-1. We will go more into detail in further lectures. But then there is a feedback loop, like most of these pathways, in which IGF activates growth hormone releasing hormone, and it activates growth hormone and it produces IGF. But also it can activate somatostatin, and obviously this has an impact on growth hormone. So what you can see here in red is positive activation, and also there are negative activation on the hypothalamic neurons. So a lot of growth hormone activates IGF, but at the same time it inhibits, right, the production in the hypothalamus of growth hormone releasing hormone, and at the same time it in the pituitary the production of growth hormone. So we will enter a little bit more into, when we do the different axes, into the negative and positive feedback loops. So this is just to show you that levels of hormones on itself, they have a negative regulation on their own production or in the somatostatin pathway. At the moment, when we do the growth hormone axis, we will enter in the biochemistry of each one of these feedback loops. So the secretion of growth hormone has a pulse during the day and is under the control, positive control of somatostatin. We can see here, right? So if we go into the other slide, you can see, right? Let me put back the other slide. This is your summary slide. So somatostatin has a negative effect on growth hormone. So the more somatostatin we have, the less growth hormone. It also, somatostatin has a negative effect on TSH, right? So remember this, high levels of somatostatin will inhibit the growth hormone production, right? And this is what you can see if you look at the secretion of somatostatin. And you can see high peak, low peak. When there is low peak of somatostatin, there is an increase of GHRH and a peak of growth hormone. Then there is another peak of somatostatin, lower peak of somatostatin, and is reciprocally inverse, right? So somatostatin inhibits the production of growth hormone releasing hormone, and then you don't have growth hormone production. If you look through the day, when we say there is pulsatile levels, there is an increase during the day, and then a reduction and another peak. So there are different peaks of distribution of growth hormone through the day. And there is a bimodal also of male and female. These peaks are different between male and female. So this is what we call pulsatile hormone release. So it's pulsatile because runs in pulses, right? And this is controlled through somatostatin in the hypothalamus that has an effect on growth hormone releasing hormone, and then will activate in the pituitary gland. So what is the role of growth hormone releasing hormone? We could say that has 4 messages for one molecule, right? And what it does, the final goal, is to stimulate the growth hormone release from the somatotroph cells in the anterior pituitary, right? But actually, it does three different roles. So it increases growth hormone synthesis, right? So it produces more growth hormone on the cells. It increases the number of somatotrophs, or cells that produce growth hormone, and it promotes cell clusters which are coordinated to produce an excess of growth hormone. So that's why it has different physiological functions. But the ultimate goal is to stimulate this secretion of growth hormone into the circulation. As I said, it has peaks during the day. And basically this graph, what it shows is that the normal levels in males and females. We can see in the axis growth hormone micrograms per liter, and these are the times of the day. And what you can see is that there is peaks in male, and then in males there is a very big peak of growth hormone, right, at this time, whereas in females this peak does not exist, right? So there are peaks through the day, but smaller. But in males, right, early in the morning there is a big increase on growth hormone production. So looking about the genetics, right, you will be familiar on dwarfism, right? So dwarfism sometimes runs in families, and the genes that code for genetic dwarfism were not understood. So with the help of genetics and using human genetics, for example, in this family was consanguineous family with Pakistani origin, and what you can see is that there are two young siblings, right, compared to the father that didn't grow, right, and were not growing and they were dwarf. So a mutation in the hormone releasing hormone receptor was identified. This was published in Nature Genetics and was a milestone in the area of genetics of endocrine conditions. This mutation, what it does is that the cell that has the receptor cannot be activated because has a mutation and therefore does not respond to the growth hormone releasing hormone. So the first mutation was identified, but now we know that there are multiple mutations in this gene. Just one second. So a knockout also has been done, and you can see here it's called the little mouse. And basically, the homozygote compared to the heterozygote, you can see that the mouse is little, and that there is mutations without the receptor, right? The mouse cannot grow because the growth hormone axis is disrupted. Here you can see different mutations that they have, like in the mouse, the little mouse, there is an aspergine to the glycine here in green, but now there is a lot of other mutations in the receptor of the growth hormone receptor, both in humans and other animals models, right? So this is very important because families that are carriers for mutations can be diagnosed a lot faster and obviously treated with the growth hormone replacement therapy to avoid severe dwarfism. So genetic panels exist in all hospitals and is first line of diagnosis, right, for the identification of genetic dwarfism. So since the first identification of the first mutations, lots of different mutations in lots of different families have been identified since that, and these are different mutations identified in lots of different families, and that has been published, right? Now, also there is another hormone releasing hormone, which is the CRH, the corticotrophin releasing hormone. The corticotrophin releasing hormone is important for the hypothalamic pituitary adrenal axis, and we will cover this in detail in the lecture of next week. But basically, to summarize very shortly, the hypothalamus produces corticotrophin-releasing hormone, which will go into the anterior pituitary, activate the corticotroph cells to produce ACTH. ACTH in the circulation will activate cortisol in the adrenal glands, and these high levels of cortisol will have an inhibitory effect feedback in the production of CRH and also arginine vasopressin, right? So the more cortisol is produced, it tells the brain not to produce so much CRH, less ACTH. So this is an auto-regulatory loop. As you will see next week, deficiencies in cortisol level produce two types of conditions. One are Cushing's syndrome or Addison. Cushing's is where too much cortisol is produced, and Addison's where less cortisol is produced. This is extremely important because the adrenal gland, as you will see next week, regulates also lots of different functions such as stress response and also inflammatory processes and other important homeostatic functions, and it can cause death if untreated. So cortisol levels are very important to be regulated. How about the next hormone? So the next hormone is somatostatin hormone, right? And this is a peptide hormone. Again, you have the sequence here, so you don't need to know obviously the whole sequence is just as an example for you to just see. So it's also called growth hormone inhibitory hormone. As we have shown in the other slide, somatostatin does not activate growth hormone but inhibits. If we go back to that slide, let me put you that slide just for you to remember, right? Here we have somatostatin. There are two hormones in the hypothalamus that have inhibitory effect, somatostatin and dopamine, okay? So this is a very useful summary slide. So if we go back into somatostatin, so it's growth hormone inhibitory hormone. It has 5 isoforms of receptors, SST receptors, and they are also GPCR, some of which can inhibit cyclic MP production. It acts as a potent inhibitor of growth hormone and also thyroid stimulated hormone. And its use, as you will see on Friday, somatostatin analogs for pituitary tumors that actually produce too much growth hormone, right? And one of these compounds is called octreotide, which basically is an analog, and it inhibits the production of growth hormone. Also found in cells of the pancreatic islet and the mucosa and the C cells of the thyroid. So the somatostatin receptors, there are several ones, there is 5 of them, and they are transmembrane, and they are GPR and GPR receptors. So the other factor is the thyrotrophin releasing hormone, or TRH. And as its name says, right, the nomenclature is quite easy, right? Because as you can see, the releasing hormone has the name of the hormone that activates. So this is easy for you to remember. So thyrotrophin releasing hormone activates the tyrosine stimulating hormone from the hypothalamus. So it's a very small peptide, right? As you can see here. It's produced by the thyrotrophin releasing hormone neurons on the hypothalamus, and it activates uh, through the tyrosine releasing hormone receptor, which is another GPCR, in the pituitary, right? And it increases phosphoinosatides and C production. It's a key regulator of thyroid stimulated hormone production in the pituitary gland that in turns will activate the thyroid. So what about, yes, and the thyroid, that as you will see from the lectures of Peter McCormack next week, right, is very important on the regulation of thyroxine and the metabolism, right? So, and you will cover also the effects of hypothyroidism and hyperthyroidism. So what about dopamine or DA, right? Is a very strong inhibitor of the secretion of prolactin. It's converted by tyrosine hydroxylases and dopa decarboxylates activity. And the DA receptor, there are two, DA receptor 1, DA2, and are again GPCR receptors and are coupled to multiple G proteins. It has a tonic inhibitor of prolactin secretion in the pituitary. So the pituitary gland would always produce prolactin unless there is dopamine. But because dopamine is constantly inhibiting the production of prolactin, then prolactin is not produced or at very low levels. What happens if we don't have dopamine? Because there is drug treatment, because there is antidepressant that work in the dopaminergic system, or for example, there is a tumor, then prolactin will be produced in the pituitary gland, and more prolactin has an effect on the production of milk by the breast. So what about the corticotrophin releasing hormone or CRH? So it's a peptide hormone, right, produced by the neurons of the hypothalamus. The CRH neurons are found in the paraventricular nucleus or PVN that we studied in the previous lecture. It's again the acts in the pituitary gland through binding the CRH receptor, which is a GPCR that stimulates CMP production and generates the production of ACTH by the corticotroph cells of the pituitary, which will release ACTH into the bloodstream, which will affect the adrenal. Is a key regulator of stress response, and mutations in CRH result in impaired stress response and also epilepsy. Just as a last slide for the also the neurons of the hypothalamus, the production of GnRH is also regulated by another neurohormone, which is called kisspeptin. And kisspeptin is a regulator of gonadotrophin releasing hormone, and it is expressed in the PVN, in the arcuate nucleus, right? And kisspeptin is shown to activate gonadotrophin releasing hormone. The gonadotrophin releasing hormone activates LH and FSH. LH and FSH in turn, right, will work on the gonad to produce gonad steroids, and high levels will have an inhibitory effect, right? We will study this mechanism in more detail at the end of the module in May when we study the hypothalamic pituitary gonadal axis. So this is the sequence of kisspeptin, and also as the same as the GnRH, is very, 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 very conserved among lots of different species. You can see here the protein sequence, and we can see human, chimpanzee, macaque, pig, cattle, horse, sheep, mouse, rat, and we can see that the sequence is all the way to zebrafish and goldfish, very, very, very similar, if not like very conserved between evolution, indicating that the hormones that regulate the reproduction have been conserved um, through evolution. There is kisspeptin 1 and kisspeptin 2, and also kisspeptin 2 is highly conserved through evolution. So again, like the GnRH, kisspeptin, and others, has a fluctuation, right? So there is increase of kisspeptin, which basically parallel the production of gonadotrophic releasing hormone and LH. As you will understand more at the end of the last module, this has a peak before birth in embryonic development, right? In the maturation and the formation of the gonads. But then it stays in a off state with not much activity, but it kicks off again in puberty, right? So this is the time life, okay? And then there is an increase in the arcuate, kisspeptin, massive increase, which will obviously produce the pubertal maturation of sexual organs, testes and reproductive and ovaries, and then this correlates with increased GnRH and LH, and then during fertile period it remains pulsatile, as you can see here. We will cover this in the fertility lectures, which I will give you at the end of in May. So then at the end of each lecture, I will give you few questions. If I was in Nanchang, right, we will cover this with clickers, but because you don't have clickers at the moment, what we will do is I will read these questions, and then you guys will think about them, and then in 1 or 2 days I will upload the answers of these questions in QM+. So then you guys can assess how well is self-assessment for you to see if you have understood the lecture. At the end of each lecture, I will read the questions, and then you can take them with you and read them, and then I will upload the questions. So one of the questions is the posterior gland, number 4 here, is that the posterior gland, the posterior pituitary, true or false? I'm not saying the answer, you guys are going to think about it and do it at your own time. The hypothesis lies in the median eminence, third ventricle, sella turcica, in front of the optic chiasm, or none of the above. The hypothalamus connects to the pituitary gland via the pars intermedia, infundibulum, median eminence, optic chiasm, or third ventricle. The pituitary gland is known as the master of the endocrine glands. However, it's controlled by which of the following endocrine glands? The thalamus, the pineal gland, the hypothalamus, the thymus, or the thyroid. Growth hormone releasing hormone receptor is steroid hormone receptor, ligand-gated ion channel, tyrosine kinase receptor, G protein coupled receptor, o tyrosine kinase associated receptor. Thyroid stimulated hormone is produced by the hypothalamus, adenohypophysis, neurohypophysis, thyroid gland, parathyroid gland. So, with all this, I want you to read the questions. Perhaps what you can do is to leave one day, right? And then try to answer these questions and see in QM Plus the right answer and see how well have you done to understand the lecture. This is only self-assessment for you. Next lecture, we will cover the hormones of the anterior pituitary gland, and that will be tomorrow. So with no further delays, we have finished the lecture today, and we have covered the anatomy of the hypothalamus and the pituitary gland. We have also covered also the anatomy of the anterior pituitary gland, the posterior pituitary gland, the hypothalamus. We have covered all the hormone releasing hormones, and we have understood that these hormone releasing hormones act in the anterior pituitary to produce further hormones that will be secreted into the circulation, and as we will see in other lectures, they will have an effect in each particular organ, forming a different axis: hypothalamic pituitary adrenal axis, hypothalamic pituitary thyroid axis, hypothalamic pituitary gonadal axis, so on and so forth. So I hope you have enjoyed the lecture, and I think we can, if you have any doubts, contact me. They need to set me up on the QQ system, but I will be able to join you, and also you can email me or use this application. So if you have questions, please let me know. And thank you very much for attending the lecture. Oh man, I don't know. Another extra hour. No.